Good morning. I'm Cristina Satriano from the University of Catania, and today I'd like to share with you the ongoing research in my lab on two-dimensional graphene-based nanomaterials with enzyme-like properties designed to combining the cancer therapy and diagnostic, which is also known as theranostics. So the graphene-based nanozymes emulate the catalytic function of superoxide dismutase, which is a class of metalloenzymes that convert two superoxide anions into triplet oxygen and hydrogen peroxide. And the sod-like activity of graphene oxide proceeds through a two-step redox mechanism. First of all, the oxidation of the carbonyl group on the graphene oxide, which is converted into another hydroxyl group, and then the second reduction step, where the hydroxyl group is oxidized back to carbonyl group. The properties that make sod-like graphene materials and especially the graphene particularly appealing in the biomedical application include the biosensing, which means that they enable the detection of key physiological markers but also many disease biomarkers, the capability to act as a broad spectrum antimicrobial agent that bypass the limitation of traditional antibiotics, and also for antioxidant therapy, neutralizing reactive oxygen species which are involved in various pathologies like inflammation, cancer, neurodegeneration, arteriosclerosis, and mimicking the natural antioxidant enzymes, the nanozymes can restore the intracellular redox homeostasis and mitigate primary and secondary oxidative damage. To note, the palladium nanoparticles also exhibit strong catalytic behavior and can combine multiple nanozyme functions, including the catalase-like activity, so producing oxygen bubble which affect the cellular mobility, the sod-like activity that we have already described, and the glutathione depletion via surface chemisorption. And when integrated with the plasmonic nanoparticles, the graphene can enhance their photothermal, photocatalytic properties, which paves the way for advanced theranostic platform that unified all these multiple functionalities. So to control the surface termination and the defect density of the graphene nanomaterials, as well as to achieve a narrow lateral size distribution of around 200 nanometers in our nanosheets, we perform a mechanical exfoliation using 2-hour ultrasonication. Also, it has to be noted that the graphene oxide is synthesized via the Hammers method, which yields functionalized graphene sheets, often coated with tightly bound oxidative debris. Then we did the base washing treatments to obtain the hydrophilic base washed GO, which is further functionalized by treatment with the thiourea bromidic acid at high temperature to obtain the RGOSH, reduced thiolate GO, introducing thiol groups and restoring the partial conductivity through reduction. Since this becomes again hydrophobic as the graphene, we need to improve the dispersibility of and complete the cycle with 2 hours ultrasonication, which helps to resuspend the nanosheets in solution and maintain a stable dispersion. Following a green and bio-inspired approach, we synthesize the palladium nanoparticles using glucose as the sole reducing agent. In a typical synthesis, the aqueous solution of the palladium precursor are added with polyvinyl pyrrolidone and glucose, then mixed together. When the pH is adjusted by the addition of sodium hydroxide, the change of the color is indicative of the formation of the colloidal nanoparticles. Same reaction, just with the introduction of the graphene family nanomaterial is done to prepare the hybrids. To optimize the fabrication of our graphene palladium hybrid nanomaterials, we monitored their morphology using electron microscopy and atomic force microscopy, and this analysis revealed a clear tendency for aggregation and a significant surface roughening for the reduced GO. We also examined the electronic properties from the optical spectra collected with the UV visible spectroscopy. In the pristine graphene, we observed the characteristic absorption peak at 230 nanometers, which correspond to the pi-pi star transition of the conjugated carbon bonds in the backbone, and we can follow a progressive red shift and broadening at a decrease in intensity going to the base wash GO and then to the RGO-SH, and with a concurrent change in the bands in the shoulder around 300 nanometer, which is the n-pi star absorption. Which also changed the relative intensity compared to the peak from 50% for the base washed and for the pristine and base washed GO to 70% for the reduced thiolated GO. This alteration, these spectral changes can be related to two main factors: the formation of nanometer size sp2 clusters and alteration in the linked chromophores such as carbonyl or CO instead of CS bonds. What is more, the pronounced bathochromic shift to 282 or 283 nanometers that we observe for the reduced GO thiolated suggests the restoration of the pi aromatic domain and the almost disappearance of this band is very well evident, as also paralleled by the comparison with the theoretical spectra calculated that we can see on the right hand side of the slides. To tune the performance of our palladium nanoparticles, we adjust the palladium to glucose ratio, producing two populations: the palladium so-called A, smaller particles, and palladium B, with larger particles. The formation was monitored by UV visible spectroscopy. Initially we can see this green line with the spectrum showing a peak at around 420 nanometers, and this is due to the ligand to metal charge transfer in the palladium complex. And as the reaction progresses, the solution changes color from yellow to dark brown, and this corresponds also to the disappearance of this band and the appearance of the plasmonic peak in the UV around 270. In the table, we can follow the red shift for the palladium particles formed in the presence of the base wash GO, and the blue shift, which means also smaller size as calculated in this optical diameter value, which is obtained according to the equation provided here. So a blue shift, smaller particles compared to the particles alone when the synthesis is performed in the presence of the reduced GOSH. And the DLS confirmed the size differences and the trends, and also the zeta potential analysis show that the hybrids of the palladium A are more similar, retain somehow the negative charge of the graphene substrates, while the palladium B hybrids reflect the palladium B profile with almost zero charge measured at the surface. And the atomic force microscopy also confirmed the presence of these very teeny particles, almost quantum dots, decorating the surface of the graphenes. Let's take a closer look at the size and the shape characteristic of the particles. Starting with the palladium A samples, we can see that the ferrite diameter ranges from 2.7 to 3.6 nanometers, with going from the particles alone to the particles prepared in the presence of the reduced graphene oxide. And the circularity values indicates more spherical particles, and also the aspect ratio point in that direction. On the other hand, the palladium B alone, that compared to the palladium B hybrids with graphene, show a trend with more irregular shapes, and also the aspect ratio, which is up to a value of 2, pointed to an elongated structure. The XPS analysis revealed that the palladium 3D spectrum includes both metallic and oxidized palladium, with the peaks at 335.6 and 337.1 electron volts from the deconvolution of the 3D 5/2 component. The surface is about 70% palladium oxide, which increased to 90% in the hybrids, which indicates more oxidation, and XRD confirm showing peaks at 31.6 and 45.4 for the palladium oxide planes 200, 220, and also the peak at 39.1 degrees for the metallic palladium 111 crystal plane. The Raman and the 2D mapping identify palladium nanoparticles as the bright spots due to the electronic structure, the plasmonic properties, and the graphene oxide sheets as brown region. All the samples show the characteristic D band at 1340 inverse centimeters and the G band at 1580 for inverse centimeter, with no observed peak shift, but instead what is changing is the integrated intensity ratio between the peaks of the disorder to the order, which points to a more disordered structure in the presence, and confirming in the presence of the palladium nanoparticles, which confirm the linking, the anchoring of the particles. This trend is also confirmed by the quantum mechanical Raman spectra calculation performed on the model systems using a Gaussian 16 model built with 2.54 times 1.82 square nanometers structure of graphene oxide and palladium nanoparticles with a diameter of 1 nanometer. Then let's move to the functional characterization of our palladium GO hybrids, focusing on their sod-like activity. We use the Friedowich method, which is a colorimetric assay that employs xanthine oxidase system to generate superoxide anions. These radicals reduce cytochrome C3 to cytochrome C2, which absorb light at 550 nanometers. The SOD and SOD-like nanozymes slow the reaction by scavenging superoxide radicals, so causing a decrease in the absorbance over time. The SOD activity is measured by how much the sample inhibits this reduction. The concentration causing the 50% inhibition is called IC50. In our results, the pristine and base wash GO have IC50 values of around 20 micrograms per milliliters. The thiolated GO has a IC50 of 60 micrograms per milliliters, and for the hybrids we see a clear enhancement. For example, the palladium B combined with the RGOSH lowers the IC50 dramatically to 0.3 micrograms per milliliter, which means 3 times more active than the palladium alone. Our hypothesis here is that the nanozyme mainly act to adsorption activation and electron transfer driven by the unpaired electrons and the multivalent surface ions, and the exact mechanism that can be further investigated depends on factors like the pH, the structure, the surface chemistry, which opens to plenty of possibility. Also, thanks to the plasmonic tuning, the thermographic analysis confirm a significant temperature rise in the palladium GO hybrids under laser irradiation, demonstrating a synergistic effects behind the sum of the single components. Heat generation was significantly higher than for either palladium and the GO alone. Also, the graphene's high surface area enhanced the proton adsorption via electrostatic interactions, increased the local reactant concentration, and accelerate the photocatalytic H2 evolution, a process which involves 3 steps. First, light absorption to generate electron-hole pairs, then the charge separation and migration, and the water splitting at the surface of the nanoparticle. So single semiconductors struggle with the rapid electron-hole recombination, which limits their efficiency. And here instead we can see a clear synergy with palladium alone gives 20 enhancement over titania, and palladium GO hybrids reaching a 40 times enhancement, showing a strong combined effect by the single components alone. And here we can see the cell viability results on PC3 cells, which is a prostate cancer line known for its potential to metastasize into the brain. And to better understand the metastatic behavior, we compared the response in PC3 with the blood-brain barrier model using GP8 rat brain endothelial cells. In the cancer model, we observe a consistent about 20% decrease in viability across all the treatment doses, not just the highest concentration, for all the hybrids as well as for the RGOSH. In contrast, for the BBB model, the graphene-based material show an enhancement in viability, while the hybrid of the palladium with reduced GO show a real toxicity. Therefore, this indicate a selective toxicity profile that can be relevant for target therapeutic applications. As to the mitosox assay, it reveals increased mitochondrial ROS production, and this rise is especially relevant in cancer cells where this elevated oxidized species can trigger apoptosis via oxidative stress pathways. And the confocal microscopy imaging confirmed the cellular uptake and the localization of the nanomaterials within a cellular compartment. And the degree of organelle disruption varied with the treatment conditions, which is also very promising for their dual use, both in terms of therapeutic impact as a diagnostic potential, especially for the hybrids. In conclusion, we developed the green synthesis of hybrid nanoparticle graphene-based nanozymes featuring eco-friendly synthesis, tunable characteristics, and also multifunctional properties. Next step include fine-tuning of the catalytic and multi-enzyme activities, as well as advancing the studies toward clinical translation. Finally, I would like to acknowledge the collaborator to this work from my laboratory at the University of Catania and other departments at the University of Catania and the University of Basilicata, and also the founding. And thank you all, and I'm ready to answer your questions.